Hi everyone, welcome to Biochem 302. This is set 8 on the fate of carbon skeletons amino acid catabolism. The suggested readings are in Leninger here from the 5th, 6th, and 7th edition, as well as the practice problems being listed here. In this set of notes, we're going to go over glucogenic versus ketogenic amino acids, the catabolism of glucogenic amino acids, phenylalanine and tyrosine catabolism, plus the catabolism of other amino acids. So let's start off by talking about glucogenic versus ketogenic amino acids. So all 20 amino acids, their carbon skeletons get catabolized to 7 major products. And these 7 major products include acetoacetyl-CoA and acetyl-CoA as point number 1 and number 2. Now, these two compounds are considered to be ketogenic. So if you degrade the carbon skeletons from the amino acids and they arrive at any of these two products, they are called ketogenic. Okay? And why is that the case? And this is because remember that most of the time if you have like an excess of amino acids being catabolized during starvation mode, this acetyl-CoA is not going to be able to enter into the TCA cycle. This is because of the loss of oxaloacetate, if you remember from set 3. Okay, oxaloacetate is being siphoned away, so this acetyl-CoA has nowhere to go. So it's most likely during starvation mode, which is often the time where you're going to be catabolizing all these amino acids. This acetyl-CoA is going to go on to make ketone bodies. Okay, and acetyl-CoA is basically the same as acetyl-CoA. It's just two acetyl-CoAs basically stuck together. So once again, this will only be broken down into two acetyl-CoAs, which will go on to make ketone bodies eventually. All right. So these compounds too, so these amino acids can also be broken down into either pyruvate, oxaloacetate, fumarate, succinyl-CoA, or alpha-ketoglutarate. And so all of these compounds are located over on this part of the diagram. Okay, so you'll notice that oxaloacetate, fumarate, succinyl-CoA, and alpha-ketoglutarate, those are all TCA cycle intermediates. So if all these carbon skeletons of all of these different amino acids are being converted into something that can turn into a TCA cycle intermediate, what can this do? This can all regenerate oxaloacetate because it can all go towards the end of the citric acid cycle, which can go on to make glucose. Right? Remember that oxaloacetate is one of the lead compounds that gets siphoned away from the citric acid cycle to go on to make glucose, okay? And pyruvate as well. So all these amino acids can be catabolized into pyruvate, and this pyruvate can be converted to oxaloacetate, which can go on to make glucose through gluconeogenesis. So the main difference is that the carbon skeletons of amino acids can either be ketogenic, meaning they have the potential to generate ketones or ketone bodies, or they have the potential to generate glucose, which means that they're going to eventually be shifted into oxaloacetate to go on to generate glucose through gluconeogenesis. Okay, so let's answer question 1. So amino acids are referred to as ketogenic. Okay, so this is if the carbon skeleton of an amino acid is converted to either acetyl-CoA or acetoacetyl-CoA. Okay, so any of those two compounds, and they have the potential to form ketone bodies under starvation conditions. Okay, so remember, anything that gets degraded into either acetyl-CoA or acetyl-acetyl-CoA are going to go on to potentially produce ketone bodies under starvation mode, okay? And remember that acetyl-CoA cannot go on to produce a net amount of glucose, right? Unless you're a plant. And we're not plants, okay? So acetyl-CoA can never be used to produce glucose. We'll write this down again. That remember that acetyl-CoA cannot be used to make glucose. Okay, and we'll just write this now. Unless you're a plant and you do the glyoxylate cycle. And we're not a plant, at least the last time I checked. All right, so that's ketogenic. Glucogenic means that all of these are going to be converted into either TCA cycle intermediate or pyruvate, which will allow you to produce glucose. Okay, so we call them glucogenic. Okay, so this is if the carbon skeleton has the potential to form oxaloacetate, fumarate, succinyl-CoA, alpha-ketoglutarate, or pyruvate. Okay, so all of these compounds that I listed under here and I'm underlining, so all underlying compounds or molecules have potential to produce glucose via gluconeogenesis. Okay, so that's how we classify amino acids. The carbon skeletons either get broken down into ones that can make glucose or they're going to make ketone bodies. And we're going to find out that some can also do both. Okay, so for example, tryptophan, this is not a mistake. A lot of students read this diagram, they're like, oh, does this mean the textbook doesn't know what it's talking about? No, it just means that because this amino acid is so large, like tryptophan, parts of the amino acid can be broken down into acetyl-CoA, which have the potential to make ketone bodies, but also some of the carbon skeleton has potential to make pyruvate, which will go on to make oxaloacetate to make glucose. It'll get siphoned out of the TCA cycle to make glucose. Okay, so I hope that makes sense to you. I do not want you to memorize this entire diagram because it is extremely difficult to remember all of this, okay? So what we're going to do is going to break it down instead into things that you already know and then add in a few compounds that I want you to specifically know for the midterm exam, okay? So there's 2 amino acids that are exclusively ketogenic. So if we look at our list over here, leucine and lysine, these are exclusively ketogenic, which means that they only go into this like blue box over here, okay? They only have the potential to make acetyl-CoA and acetyl-CoA, which go on to make ketone bodies, okay? So remember, leucine and lysine are losers because they all start with L. Okay, so leucine and lysine are losers. You will never be able to get a net gain of oxaloacetate from leucine or lysine. Okay, so remember, those are ketogenic, and I want you to know this really well, so make sure you highlight this for studying on your midterm exam. Okay, 13 amino acids are classified as exclusively glucogenic. Okay, so it has the potential to make glucose. So this includes alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamate, glycine, histidine, methionine, proline, serine, and valine. And that's a lot. Okay, and I don't want you to know that. So the ones that you don't need to know are the routes that are in red. Okay. So no. The only ones that I want you to know are the ones that are in blue. So that's alanine, asparagine, aspartate, glutamine, and glutamate. And guess what? We already went over all of these in other parts of the course already in set 6 and set 7. So that's why I put them in here, okay? So these amino acids have carbon skeletons that can all produce, for example, pyruvate, oxaloacetate, or alpha-ketoglutarate. Okay, so those are glucogenic. And then we have 5 amino acids that are classified as both. So this includes isoleucine, phenylalanine, tryptophan, and tyrosine, okay? And we're going to cover these ones that are in blue, a little bit later in the set of notes. So I know you're probably looking at this now and be like, we didn't go over this before, but we will cover this by the end of the set of notes, okay? but you don't need to know threonine. So remember that they're classified either as ketogenic, glucogenic, or they can be both ketogenic or glucogenic. And if there is a question that asks you about amino acid that's in red, I will give you the information and the pathway on an exam, and you will have to tell me whether or not it's glucogenic or ketogenic just by looking at what end products are made when this thing gets catabolized. Okay, so there could be another example on your test later on that's in the red, but again, I will give you the information, so you're not expected to like look it up or anything. I'll put it there, and you just have to look at what compounds are made, and we'll go over one example a little bit later. Okay. So this table in your notes is something that we're going to fill out as we go along, because every amino acid that we're going to work with here in the left column is going to give you a slightly different ATP gain when you catabolize it. So just like we did in beta oxidation, we kind of went over like how odd number chains and even number chains and double bonds all gave you kind of different ATP values. We're going to do the same thing and just summarize it in this table and calculate the net ATP gain here at the end. Okay, so to go over this table, we need to understand what's going to happen. So this is a TCA cycle diagram that again is showing you the same thing that you learned about in set 2, because we're going to see a lot of the carbon skeletons go back into the TCA cycle, and we're going to have to see where we left off and see how much ATP gets generated. Okay, so let's go through all these step by step, and then we'll refer back to the table in that diagram of the TCA cycle to calculate the ATP counts that you would get when these amino acids are completely catabolized. Okay, so we have glutamate first, and it's going to undergo oxidative deamination by glutamate dehydrogenase. Okay, so here's glutamate, NC-alpha-CO, 2 methylenes with the carboxyl group. And so we know that this reaction here, which uses water, and the enzyme glutamate dehydrogenase is going to produce alpha-ketoglutarate and NH4 plus the ammonium, which is going to go into the urease cycle, okay? And notice in this step, we produce an NADPH, and just assume for this course that NADPH that gets produced during this glutamate dehydrogenase step is going to be worth 2.5 ATP, okay? So let's fill out this table over here. So how many nitrogens do we have in glutamate? We have 1. Now, what is the ATP cost of the urea cycle? So this goes back to our set 7, okay? So let's write down, this is from set 7. And it says at the bottom of the table, right, that assume that all amino groups enter the urea cycle through CPS1 and that aspartate was abundant, right? So if we assume those two things, that means we are going to have to calculate some of the cost and gain of the ATP in the urea cycle, okay? So let's just go over that really briefly, right? So if we have all amino acid groups going to the urea cycle through CPS1 and aspartate, remember that we're going to have the reaction of CPS1, which is the committed step there of the urea cycle, so that uses minus 2 ATP. Then we have argininosuccinate synthetase, which also uses minus 2 ATP. And then as the urea cycle goes on, we're going to produce a fumarate. Okay, so we're going to write down that fumarate gets produced, which is going to give us plus 2.5 ATP. So in total, this is going to give us minus 1.5 ATP per nitrogen to the urea cycle. Okay, and once again, this is assuming that CPS1 and aspartate are abundant, okay? So let's just write this down here, okay? So if we have 1 nitrogen going to the urea cycle from glutamate, the cost of the ATP in the urea cycle is going to be minus 1.5. Okay, minus 1.5. Remember that we're gaining 2.5 ATP from this glutamate dehydrogenase, so that's plus 2.5. Okay, and now this alpha-ketoglutarate is going to go to the TCA cycle. So when alpha-ketoglutarate goes to the TCA cycle, it enters right over here. So we get 2.5 ATP plus 1 plus 1.5 plus 2.5. And we stop at oxaloacetate always, just like we did in set 2 with beta oxidation, okay? So that's 3.5, right? That's 5, and then that's 7.5. So we're going to get plus 7.5. Whoops, that's the wrong place to write that. This is alpha ketoglutarate is the carbon skeleton, and we're going to get plus 7.5. So when we add up the net amount of 80 ATP that we get there, in that column, we're going to get 8.5 ATP. So plus 8.5 ATP equivalents. Every time we put a glutamate going to the urea cycle and oxidizing its carbon skeleton. Okay, so that's glutamate. Let's try glutamine. So glutamine, remember, goes and arrives at the liver and undergoes hydrolysis by the enzyme glutaminase. So here's glutamine and there's glutaminase. This is a hydrolysis reaction. So we're using water to split off this ammonia. Okay, so this amino group is going to leave. Remember, this is a side chain amino group at this point, right? So this is going to go directly into the urea cycle. Okay, and then we're left with glutamate. So this is just the same as before. We're going to take our glutamate, which is left over, which still has a nitrogen on the alpha amino group, and it's going to undergo the glutamate dehydrogenase reaction, which is going to pull off the amino group to go in the urea cycle. And then we're left with the carbon skeleton of the alpha ketoglutarate once again. And remember that we gained this 2.5 ATP from this NADPH, okay? So remember, this is the side chain amino group, and this is the alpha amino group that's going to go to the urea cycle. Okay, so the number of nitrogens that are going to the urea cycle is going to be 2 for glutamine. Let's write this down, 2. Okay, so because there's 2 nitrogens going to the urea cycle and there's 1.5 each, so there's 1 and 2, it's going to cost us 3 ATP by sending those nitrogens to the urea cycle. We had to use an oxidative deamination step here, which is going to be plus 2.5. And the final carbon skeleton that is made, again, is going to be the same, alpha ketoglutarate. And we're not going to go over this again. When you put alpha ketoglutarate going to the TCA cycle, it's going to yield 7.5 ATP. So when we add up this column, it's going to be plus 7 ATP equivalents. The reason why it's 1.5 less than glutamate is because of the number of nitrogens, right? We have 1 and 2 there. Just that extra nitrogen costs the cell a little bit more to get rid of it through the urea cycle. Okay, so that's glutamate and glutamine. Let's go to aspartate. So what aspartate is going to do is it's going to undergo an aminotransferase reaction. So we've seen this before. This is AST, aspartate aminotransferase. It's going to transfer the amino group onto alpha ketoglutarate to make glutamate, and then the carbon skeleton that's left over is oxaloacetate. Okay, this glutamate that gets produced here is going to undergo oxidative deamination to release the ammonia to go into the urea cycle. But however, we cannot count this alpha ketoglutarate as part of the original carbon skeleton of aspartate, okay? It was simply used as a vehicle to bring this nitrogen into the urea cycle, okay? So these carbons don't actually come originally from the carbon skeleton of aspartate, okay? And that's why if we were to write out the overall reaction, this would cancel out and this would cancel out here, okay? So we can't really count that. So let's go back to our table. Aspartate only has 1 nitrogen, which means it's going to cost 1.5 ATP to go through the urea cycle, assuming we're going through CPS1 and that aspartate is abundant. We're going to see oxidative deamination produce 2.5 ATP over here. And this time, our final carbon skeleton is not alpha ketoglutarate. It's actually oxaloacetate. Remember, that is the alpha keto acid of aspartate. So this is going to be oxaloacetate. And by convention, oxaloacetate is not actually worth anything in the TCA cycle because we go back. That's where we usually have to stop. Now, that doesn't mean the oxaloacetate isn't worth anything in the cell. It's actually important because, it could come back to regenerate citrate by bringing in an acetyl-CoA, or it could go on to make glucose through gluconeogenesis, okay? So even though net-wise the ATP counts look low, producing oxaloacetate still has its benefits. Okay, so we're going to add this up, and it should be plus 1 ATP equivalent. for aspartate. Okay, let's try asparagine, which I call the Asian amino acid because it's ASN is the abbreviation. So the difference between aspartate and asparagine is that there's an extra nitrogen there on the side chain. So just like glutaminase, we're going to have an enzyme here called asparaginase, which is going to use a hydrolysis reaction to remove this nitrogen from the side chain. And this nitrogen will go directly into the urea cycle. Then aspartate is just going to be like the exact same thing we saw on the last one. So it's going to undergo the aminotransferase reaction through aspartate aminotransferase. It's going to transfer the amino group onto alpha ketoglutarate to make glutamate. And then the alpha keto acid of oxaloacetate is going to go to the TCA cycle, which is worth nothing. Remember, that's worth nothing in terms of ATP. The glutamate, which carries the nitrogen from the aspartate, is going to undergo hydrolysis through glutamate dehydrogenase, and it's going to produce an NADH or an NADPH there, which is worth 2.5 by our convention. We do not count the carbon skeletons from alpha ketoglutarate again because this is not part of the original carbon skeleton from asparagine. And then the remaining nitrogen, which gets taken off here, is going to go into the urea cycle. So let's summarize this in the table. With asparagine, we used 2 nitrogens there, or we have 2 nitrogens on the structure, so it's going to be -3. We still have to do an oxidative deamination step, which occurred right here. It's going to be worth 2.5. Remember that the carbon skeleton that gets produced from asparagine is the same one that gets produced from aspartate, which is going to be oxaloacetate. And by our convention, it's not worth any ATP. When we add up all of these values, we are going to get minus 0.5 ATP equivalents. This again does not mean that when we break down asparagine when our body's under starvation mode, that it's not worth anything. Remember that oxaloacetate, even though by our convention produces no ATP, it can still go on to produce glucose through gluconeogenesis, or it can further go on to make citrate with an addition of an acetyl-CoA. All right, so that's the second last column of our table, or the second last row of our table. The last of the glucogenic amino acids we're going to cover is alanine. So alanine, look at this, is pretty easy structure. NC-alpha-CO, methyl group here is the R group. And we're going to undergo ALT, which is the alanine aminotransferase enzyme. This means the amino group is going to get transferred onto alpha ketoglutarate to make glutamate, and we're going to get left over with the carbon skeleton of pyruvate. Now, if you remember from your Biology 201 and your Biochem 202, this pyruvate has one of 2 fates. It can either go through PDH, which stands for pyruvate dehydrogenase, which is going to what we're going to do in step 2A, or it can undergo carboxylation by pyruvate carboxylase, which we're going to go over in step 2B. Okay, so in step 2A, if we do this route, pyruvate is going to combine with coenzyme A. Through the pyruvate dehydrogenase complex, it produces an NADH, and you get an acetyl-CoA out of it, which is going to go into the TCA cycle. CO2 is also produced. So once you get that as one of the options, there's also this pyruvate carboxylase. Okay, we're going to go over and just write down the information first for pyruvate dehydrogenase, just so we don't confuse anyone, okay? So we're going to kind of split this row in half. So let's draw a little line here. Okay, so remember that alanine only has 1 nitrogen, so 1 nitrogen will go to the urea cycle. Okay, we're still going to eventually have a glutamate, which is going to undergo oxidative deamination. Okay, so this glutamate is going to go oxidative deamination, which produces 2.5 ATP. The carbon skeleton of alanine is pyruvate. Okay, so let's just write this down twice because it's the same. Now, in terms of the carbon skeleton and the ATP that accounts for it, depends on where this pyruvate's going to go. Is it going to undergo reaction with bicarbonate to use pyruvate carboxylase, or is it going to undergo the PDH complex, pyruvate dehydrogenase, okay? So if we use PDH, pyruvate dehydrogenase, okay, we're going to get 2.5 ATP from this NADH, and we're going to get another 10 for this acetyl-CoA over here. So this is plus 12.5. And then we're going to have pyruvate carboxylase over here, which is going to actually use an ATP. So it's going to be -1 there, okay? So if we use PDH, we're going to actually gain 13.5 ATP equivalent. And if we add up the values for this part of the table, it's going to be 0 if we use pyruvate carboxylase. Okay, but once again, even if we do pyruvate carboxylase, we produce this oxaloacetate, which is worth 0, right? But it still has value, this oxaloacetate, even if it's not just an ATP. Okay, so going back to our slide again that talked about the glucogenic amino acids that you're supposed to know, right? This includes alanine, asparagine, aspartate, glutamine, and glutamate. We now know these parts of the diagram, right? We know that alanine is going to get converted into pyruvate, okay? But this pyruvate can go directly into acetyl-CoA through PDH, or it can go through oxaloacetate here by pyruvate carboxylase. We went over those two routes. For asparagine, the alpha keto acid that gets produced is going to be oxaloacetate, so that goes directly into here. Same with aspartate, which is not worth any ATP because this is where we stop our ATP counts in the TCA cycle. And then glutamine and glutamate, the carbon skeletons that correspond to these are going to be alpha ketoglutarate, which are going to go into the TCA cycle to eventually form oxaloacetate, but we do get 7.5 ATP per alpha ketoglutarate. Okay. So I hope this summary table makes sense. And hopefully it compresses the information in a way that makes it easier to understand. Okay. So remember, those are the amino acids that you are responsible for knowing for how to generate the ATP counts when we do amino acid catabolism. Okay, in this next section of notes, we're going to go over section 7.3, which is on 11.3, which is on phenylalanine and tyrosine catabolism. So you can see phenylalanine over here. We know the structure of phenylalanine, NC-alpha-CO, has a methylene, plus it has this aromatic ring on it, which is the phenyl. Okay, so phenylalanine makes sense in terms of how it's named. Now, phenylalanine is kind of weird because when it's being catabolized by the body, like being broken down, it does not require an amino acid aminotransferase right away. So what happens here is that phenylalanine actually gets converted into tyrosine first, and then it's going to be met with an aminotransferase. But let's talk about this enzyme because it has some health implications as well, okay? So let's look at this. Catabolism of phenylalanine to tyrosine uses NADH, and it's going to be done indirectly through this cofactor that's called tetrahydrobiopterin, okay, which is THB. So you can think of this enzyme though as just using NADH. That's fine, but it does use the help of this THB there. And we'll notice that we also have to hydroxylate this ring. So we're going to use an oxygen, a diatomic oxygen here, and we're only going to use one of these oxygens plus the hydrogen ion here, and put that onto the aromatic ring. And a water molecule is actually going to get produced in the product here. Okay. So remember, we are hydroxylating this over here. Now, what does it also say here? Okay, so question 2, why is phenylalanine considered to be an essential amino acid, but tyrosine is considered to be conditionally essential? Okay, so we call phenylalanine an essential amino acid because our body has no way of making this. You have to get this in your diet. Okay, but tyrosine can be made from phenylalanine, so therefore it can be made on the condition that you do have phenylalanine. Okay, so this is question 2. Let's change the color. Okay, so phenylalanine is essential. Okay, so the human body has no method of synthesizing it. And then tyrosine is conditionally essential. And this is that the body can synthesize if and only if phenylalanine is already abundant. So if you ingest phenylalanine, technically you can get tyrosine, but if you don't have any phenylalanine, you don't get any phenylalanine or tyrosine. I hope that makes sense. Okay, so this just talks about the rest of the reaction there. I already mentioned this before. And yeah, you should be able to recognize that phenylalanine and tyrosine, those structures, because those are 2 of the 20 amino acids that are important. Okay, this diagram is just showing you an FYI here. This is for your interest. It's just showing you how NADH is going to donate 2 electrons to dihydrobiopterin, which is the quinoid form, to form the tetrahydrobiopterin. This leaves as the oxidized NAD+, and then these 2 electrons are taken once again to be donated to phenylalanine to help contribute to the hydroxylation of that aromatic ring to make tyrosine, okay? So, again, you don't need to know this really. It's just showing you again that that NADH is used as the original source of electrons, but it does go through this dihydrobiopterin and tetrahydrobiopterin intermediate, and I will not ask you about these structures on an exam, okay? So that's it. So this again has some health implications I mentioned before, right? Phenylalanine hydroxylase. If an individual doesn't have this enzyme, what happens is that it results in this condition that's called phenylketonuria, which is PKU. And I remember this name of this is like Pikachu, like Pokemon. So phenylketonuria, okay? So 8 out of every 100,000 births, there's going to be a mutational defect in this enzyme, which means that the individual who has this disease cannot convert phenylalanine into tyrosine properly. And so what happens is that this person is going to have an increased concentration of phenylalanine in their bloodstream. Okay. So let's answer these questions down here. It says tyrosine is a precursor molecule for the production of melanin, which is used to make skin pigments. So why do some individuals with PKU have fair skin and light colored hair? Okay, so individuals with PKU, they have fair skin, like it's pretty pale looking, and they have light colored hair. And that's because individuals who have this disease, they have the inability to produce tyrosine, which can then go on to make melanin. But since they can't make the tyrosine, it cannot go on to make any melanin. Okay. So question 3. So we'll just write down that PKU patients have low concentrations of tyrosine due to lack of phenylalanine hydroxylase. Okay, as a result of having low amounts of tyrosine, low amounts of melanin can be produced. And this means that you get a low amount of skin pigments being made, which is why they're fair skinned and the hair color is light. Okay. And why is having phenylalanine in the blood dangerous? Like if you have lots of quantities of phenylalanine in your blood, so this, how does increasing levels of phenylalanine in the blood affect brain development and how is PKU treated? Okay. So if you have large quantities of phenylalanine in blood, this affects brain development. And you're probably wondering why. Okay, so when your brain transports phenylalanine, or when your body transports phenylalanine into the brain, it has to cross what's called the blood brain barrier, or the BBB. And so there's a transporter there that transports hydrophobic amino acids. And phenylalanine falls under the umbrella of hydrophobic amino acids. And so what happens is that transporter gets saturated with all this phenylalanine that the brain only receives tons of phenylalanine, but it doesn't get the other hydrophobic amino acids. So when a child is developing, they're only getting phenylalanine to make the proteins that you need to develop the brain, but they're not getting, for example, like leucine or isoleucine or alanine to finish off the protein synthesis. Okay, so increase in phenylalanine in blood affects brain development. This is because phenylalanine saturates the hydrophobic amino acid transporter in the blood-brain barrier. This means the brain becomes saturated with phenylalanine but lacks other hydrophobic. Oops, I can't spell hydrophobic. Hydrophobic amino acid side chains. Okay, so i.e., brain is deficient in things like leucine, isoleucine, valine, etc. Okay, and because of this deficiency, there is lack of brain development. Okay, and how is this PKU treated? Well, PKU is treated. So PKU treated by, as you probably guessed it, a low phenylalanine diet and in general a low protein diet because proteins contain some amounts of phenylalanine, okay? And that's why a lot of health manufacturer or a lot of food manufacturers, when they're trying to distribute foods, they have to say whether or not they contain phenylalanine on them. If you have ever looked at the ingredients on a can of Diet Coke, it'll say aspartame is here, but it contains phenylalanine, and that's because that is an important thing for PKU patients. They need to know that Diet Coke contains quite a large quantity of phenylalanine in there from the aspartame. Okay, so how is PKU like how do you diagnose it? Okay, so what happens is that when you have this increase in phenylalanine, there's this aminotransferase that rarely gets used. Okay, because most people who don't have PKU, this phenylalanine never builds up. But remember, aminotransferases, they're controlled basically whether or not the reactants or products are rich in concentration. So if you have a high concentration of phenylalanine, it's going to push down this reaction towards the bottom. Okay, so what happens is that the amino group gets transferred onto pyruvate to make alanine. This is a little bit different. It's not alpha ketoglutarate. Okay, and what happens is that we get the alpha keto acid of phenylalanine, which is going to make phenylpyruvate. So individuals with PKU and they consume a lot of phenylalanine, for example, they're going to have a really high quantity of this, which will make a high quantity of this guy over here, which is phenylpyruvate. And this is used as a diagnostic activity because most healthy individuals who do not have PKU will not have high quantities of this in the urine. So, what a doctor or a lab technician can do is they can detect in the urine the presence of phenylpyruvate by adding in this compound that's called ferric chloride, which is FeCl3. And when they add this FeCl3 to a patient's urine who has PKU, it'll turn this characteristic green color, which means we're positive for the compound of phenylpyruvate. Okay, so that is what's happening over there. And that phenylpyruvate down there as well, just letting you know, can be decarboxylated to form this other compound called phenylacetate and can also be reduced to form phenylactate. But again, these aren't really the important reactions over here, just showing you that this compound can also be degraded. The important thing that I want you to know is that when you have a high quantity of phenylalanine, this rarely used aminotransferase gets put into play and it's going to make a lot of this phenylpyruvate. Okay. So, question 5. With your knowledge of the enzyme involved in the formation of phenylpyruvate, why do you think individuals who have PKU will have a buildup of phenylpyruvate? Okay. So, this is aminotransferase enzymes, right? So, let's write this down, right? This is an aminotransferase. So, aminotransferases have delta G naught prime value of 0, right? So, this means that they're heavily influenced by the concentration of reactants and products. So the direction of reaction is heavily influenced by the concentration of reactants and products. Okay, so if we have a high concentration of phenylalanine, this pushes the reaction forward to make high concentrations of this compound that's called phenyl pyruvate. Okay, so I hope that makes sense. It's just a review of our knowledge about phenylalanine and aminotransferases once again. Okay, the next thing I want to talk about is aspartame. So I mentioned this to you already before. This is a dangerous compound for individuals who have PKU. They don't want to consume a lot of this. So aspartame is a chemically modified dipeptide, and it's used as an artificial sweetener in a lot of sugar-free diet sodas like Diet Coke, for example. So suggest why aspartame may be dangerous to individuals who have PKU. So um, if you look at the structure of it, right, go N-C-alpha-CO. This is going to be the amino acid aspartate or aspartic acid. This is the side chain of aspartic acid. And then N-C-alpha-CO, this is going to be the side chain of phenylalanine. And this is a methylated C terminus. So that's aspartame. And if you're wondering how people even figured out that this would taste sweet, is that this was made in a chemistry lab where there was a peptide chemist or a protein chemist that was synthesizing peptides, and he happened to lick his gloves and figured out when they synthesized this dipeptide of aspartate and phenylalanine, it actually tasted kind of sweet on the tongue. And obviously, it's not going to be very toxic because these are just going to be broken down into two individual amino acids, which your body's very used to seeing. Okay. So, why is it dangerous to PKU patients? Well, when it goes in the body, this gets hydrolyzed by like a dipeptidase, for example, like an enzyme that's going to cut the peptide bond here. And that releases phenylalanine, and the phenylalanine will increase in concentration. And that's why on Diet Coke, you see that the ingredients is aspartame, and then in brackets it says contains phenylalanine. Okay, so let's write this down. Let me just move this table. Okay, so why is it dangerous? So this is because aspartame, I can't write. Aspartame is a dipeptide of aspartate and phenylalanine, and then when consumed, produces an increase in the amount of aspartate and increases in the amount of phenylalanine, right? And remember that if you have an increase in phenylalanine, this is dangerous to the PKU patients, the phenylketonuria patients, okay? So that's why it's dangerous, and they have to write that aspartame contains phenylalanine on their ingredients package. And look at all this now. Popular diet soda, like we talked about Diet Coke over here. Look at the ingredients in it: carbonated water, caramel color, aspartame. Like, you know what this all is, right? You know what water is, obviously. Caramel color, who knows what that is? But aspartame, you know the structure of that. You know phosphoric acid, you know the structure of that. Potassium benzoate, we learned about that as a nitrogen scavenger. Natural favors, whatever. Citric acid, you know what this is from the TCA cycle. And then caffeine, we learned about that structure too when we talked about inhibition of phosphodiesterase. So look at that. You take this course and you're only, what, 3 or 4 weeks in, and you know everything that's in Diet Coke in terms of the chemistry behind it. Okay, so that's kind of neat. All right. So let's finish off talking about this phenylalanine catabolism, right? So we know this step already. This is phenylalanine hydroxylase. So we hydroxylate the aromatic ring to make tyrosine. Now we're going to talk about how we're going to catabolize this entire thing. So tyrosine is further catabolized to this compound that's called homogentisate, which is going to take two enzymatic steps. So the first step is going to be a aminotransferase reaction. So that's nothing new here, right? We're going to take this amino group, we're going to slap it on to alpha ketoglutarate to make glutamate. And then we end up with this alpha keto acid, which is called the para-hydroxyphenylpyruvate. Okay, so if you know the structure of tyrosine, you know the structure of this guy over here. You just go OC alpha CO, and this is the side chain of tyrosine already. Okay, so that is that's a compound that gets produced after you use tyrosine aminotransferase. Okay, um, afterwards this becomes catalyzed. This reaction over here catalyzes a dioxygenase. Okay, so what's going to happen next is that they're going to try and put two hydroxyl groups on the aromatic ring instead. Okay, and this produces this compound that's called homogentisate. Okay, and I don't want you to know this enzymatic reaction. I'm not really caring about this. We're caring about this, okay? The production of this homogentisate because it does have health impacts, all right? So all I want you to know is that tyrosine undergoes an aminotransferase reaction to produce this alpha keto acid, which you don't need to know the name of or the name of. You don't need to know this. The structure you should kind of recognize because it's the alpha keto acid of tyrosine. Then it's going to undergo a dioxygenase reaction to produce homogentisate, and you don't need to know this enzyme. You don't need to know, you don't need to memorize the structure of this, but you do need to know this homogentisate because I will ask you this on a test. Okay, so make sure you know homogentisate. I'm warning you again. Okay, so this homogentisate is bad because we're going to see that if you have this enzyme here, which is called homogentisate 1,2 dioxygenase, you have no problems. But some individuals lack this enzyme, and they have this condition which is known as alkaptonuria. Okay, and that's when you get these increased large concentration of homogentisate. Okay, so individuals who lack the enzyme homogentisate 1,2 dioxygenase have this condition called alkaptonuria. Okay, they have high concentration of this, and they're going to find that they have high levels of this in their urine. Okay, oxidation of homogentisate turns the urine this characteristic black color, and it can also form these black deposits in the eye. Okay, and you can also get black deposits in ear cartilage as well. So these are sort of hallmark features of this alkaptonuria disease. So it's an extremely rare disease. It's only affects about 4 in 1 million. But it's a little bit bad because people with alkaptonuria have a higher chance of developing arthritis later on in life, and that's likely due to these deposits that are building in the joints and then causing them to become inflamed later. Okay, so they have a high risk of developing arthritis. It's not that bad in terms of all that, but it's kind of interesting because it can develop arthritis, and usually people who are even affected by this disease, they don't even realize it until they're over 30 years old. Like they just think that it's normal to have those black deposits there, but yeah, that's what causes this disease though. So you have a lack of this enzyme. Okay, so that's alkaptonuria. Now, what happens next after it goes through the dioxygenase is it produces this compound called mallyl acetoacetate, which you don't need to know. It undergoes an isomerase reaction, which means there can be some shifting of chemical bonds to form fumaryl acetoacetate. It's going to undergo a hydrolysis reaction. All of this stuff, I don't care that you know, okay? So you don't need to know this stuff. The stuff that I would care that you know about is what happens with these N compounds, okay? So we get production of fumarate, we get production of acetoacetate as well. Okay, after this thing gets split in half. And then this enzyme called beta ketoacyl CoA transferase, which we saw back in set 3, which steals the CoA from succinyl CoA to make succinate, makes this compound that's called acetoacetyl CoA. All right. So the main thing I want to show you here, okay, is that there's two compounds that are produced, right? This is fumarate. This is acetoacetate, which eventually makes acetoacetyl CoA. So now that you've seen the whole breakdown pathway of phenylalanine and tyrosine catabolism, how would you classify them in terms of what kind of amino acids are they? Are they glucogenic? Are they ketogenic? Are they both? Or are they none of the above? And so just by looking at this, right, I would say that they're both because this fumarate can go on to form oxaloacetate, which can go on to make glucose. And this acetoacetate is already a ketone body itself, but it can be added with a coenzyme A to make this compound, which you know is ketogenic, okay? And this is exactly the kind of thing I'm going to show you on your midterm. I'll show you an amino acid that you've never seen before, which is going to have some kind of breakdown reaction, which I don't expect you to know the names of the intermediates. But when I show you the end products of the pathway, you should be like, oh, yeah, I can classify this amino acid based on what is produced, right? So when you look at this, if this was an exam question, you would say, oh, I know it makes fumarate, so that means it's glucogenic. I know it makes acetoacetate and acetylacetyl-CoA, so that gives me the idea that this has to be a ketogenic amino acid as well, so it has to be both. Okay, so let's write down the answer for here. So this is going to be both. Okay, so it's glucogenic because of the fumarate produced, and it's also ketogenic because of the acetoacetyl-CoA. Okay, this is section 11.4 on the catabolism of other amino acids, and this is the summary table. So once again, we're just going to fill this out as we go along and put in the numbers to calculate the ATP gain when we catabolize these amino acids in the body. I want you to keep in mind again this little asterisk statement saying that all amino acids enter the urea cycle through CPS1 and that aspartate is abundant, okay? So because of this, okay, we're gonna again know that we have to use CPS1 over here, which is gonna be -2 ATP. We're gonna use argininosuccinate synthetase, which is -2 ATP equivalents, and we have fumarate produced, which is +2.5 ATP. So the net is gonna be -1.5 ATP per nitrogen. Okay. So let's start by talking about this diagram, okay? All this is saying is that alanine undergoes an aminotransferase reaction to produce pyruvate. So alanine aminotransferase produces pyruvate, and then pyruvate is the alpha keto acid of alanine. And we know that this pyruvate has one of two fates. It can either go through pyruvate dehydrogenase, PDH, or pyruvate carboxylase, okay? We already went over the alanine one in the first table that we did earlier. So I'm not going to go over this again, okay? So this diagram includes alanine. That's why it's just being restated here, okay? We just covered phenylalanine and tyrosine catabolism in the section 11.3, so we're going to do it now since it's fresh in our mind. Okay, so phenylalanine and tyrosine, they have 9 carbons each, and phenylalanine is first catabolized into tyrosine by phenylalanine hydroxylase. So I just want to point out that when we use phenylalanine hydroxylase, this is going to be -2.5 ATP. Okay, why is that again? Is because when we go back to that enzyme, we use an NADH. It's indirectly through tetrahydrobiopterin, but we still use an NADH here, so that's why it's -2.5 ATP, okay? So that's for phenylalanine. It gets first catabolized into tyrosine. We already know that through phenylalanine hydroxylase. That's the enzyme that is lacking in PKU patients. And let's just look at the labeled carbons that are on this diagram, right? So we see that there's 4 in the aromatic ring that are blue, and we see that those end up as fumarate. We already saw fumarate as one of the carbon skeletons that get produced in the last couple of slides, right? So these fumarate molecules are going to go to the TCA cycle to eventually generate oxaloacetates. We get some ATP out of that. And then the other 4 carbons, which are in pink, are eventually going to go on to make acetoacetate and acetoacetyl-CoA. All right. But let's do the ATP count for this, okay? So again, I don't want you to memorize this diagram because it is there's a lot of stuff in it, okay? Again, when I give you an amino acid on the test, which I'm going to give you one that you've never seen before, I'm going to show you the exact breakdown pathway, but you have to count what's in between all the steps, and you have to take into those account and then calculate the ATP gain, okay? So we're going to just do this one as practice, right? Phenylalanine and tyrosine. So phenylalanine How many nitrogens does it have going to the urea cycle? It's going to have 1. Okay, same with tyrosine. So the ATP cost, if we assume that CPS1 is being used and aspartate is abundant, is minus 1.5. Okay, when that amino group goes on to glutamate, it has to undergo oxidative deamination to produce 2.5 ATP. So this is going to be the same as we saw before. And we know that the final carbon skeleton is going to be, there's going to be 2 different ones. So in phenylalanine, we get the production of fumarate as well as acetoacetyl-CoA. And same with tyrosine. We get fumarate and we also get acetoacetyl-CoA. Now, do we classify this as glucogenic, ketogenic, or both? Remember that fumarate is glucogenic and acetoacetyl-CoA is ketogenic, so we qualify as both. Now, when fumarate goes to the TCA cycle, how many ATP do we get from it? It's going to be plus 2.5. Okay, what about acetoacetyl-CoA? This is going to be plus 20 because it's basically 2 acetyl-CoA stuck together. Okay, so we're going to produce the net from all of this. Remember that we have to take into account phenylalanine hydroxylase. So there's going to be another minus 2.5 here, but when we add it all up, let's just put in a different color here in green. Our net wise, we're going to get plus 21 ATP equivalents. Okay, so remember this phenylalanine hydroxylase only qualifies for the catabolism of phenylalanine, okay? So that is for phenylalanine. What about tyrosine, right? Tyrosine is a little bit different because we don't have, it's basically the same pathway, but we don't have phenylalanine hydroxylase. So by that account, it should be 2.5 more. So we have no phenylalanine hydroxylase here that's going to be used. And so our net is plus 23.5 ATP equivalents. Okay, so that's phenylalanine and tyrosine. We kind of saw that pathway before. We did see that pathway before, but again, the most important thing I want you to know is what are the final end products. So when you see this, you should have been like, oh yeah, it produces a fumarate, right? When you see this, it produces an acetoacetyl-CoA, which can be sliced up to make 2 acetyl-CoAs, which is 20 ATP. Okay, so if you're wondering where this additional carbon goes, it gets lost into carbon dioxide, and that's just an FYI. All right, let's do this as an example, like if this was a test question, okay? So tryptophan. So again, I don't expect you to memorize this entire pathway because it's so long. Like it even abbreviates in the textbook. There's 9 steps here, another 4, so it's quite big. But I just want you to appreciate the diagram and look at where the final carbon skeleton is going, okay? So it's showing you that tryptophan has 11 carbons and it gets catabolized through multiple enzymatic steps. 4 carbons from the tryptophan aromatic side chain go on to form acetoacetyl-CoA. So these ones that are in purple go through here, they go through here to make this glutaryl-CoA, and then form more steps to make this acetoacetyl-CoA. All right. Then we have 3 carbons from this tryptophan, which are going to go on to make pyruvate. So this pyruvate over here is going to get made, and when we had to talk about where this is going to go, if it goes through PDH or if it goes through pyruvate carboxylase. Okay, so let's go through this pathway, and we're going to go through it and write down the numbers here, okay? So tryptophan, how many nitrogens do we have? We have 2. Okay, it's going to go through the urea cycle, so it's going to be minus 3 in total. Okay, so those 2 are eventually going to go to the urea cycle. And we're going to get plus 5 for this because we're going to get 2 glutamates being produced from this. Okay, so again, I would show you on the real test that there's 2 glutamates produced, but there actually is. Okay, and the final carbon skeletons that get broken down, they're going to form into pyruvate and to acetoacetyl-CoA. So if it goes through pyruvate, goes and makes pyruvate and acetyl-acetyl-CoA, we classify this as both because pyruvate has the opportunity to make glucose through gluconeogenesis, and this is ketogenic. So if it's pyruvate, we're just going to go through the easy one. We're just going to say it's pyruvate dehydrogenase, which is going to make 12.5 ATP. Okay, and acetyl-acetyl-CoA is going to make 20 ATP. Okay, so if it uses PDH, it's going to produce 2.5 ATP. So that's from 12.5, sorry, if it's PDH, but there's also 2.5 ATP that we forgot to count in this step, which was making the from the alpha keto adipate there. Okay, so we're just going to write down this is from the alpha keto adipate. So net wise, we're going to get 37 ATP, and that kind of makes sense because this structure is really, really big in tryptophan. Okay. So I know that looked a little bit confusing. The most important things I want you to know is that just by looking at this diagram, we saw that it produced pyruvate. And I would say in the exam question that it would go through pyruvate dehydrogenase. And the other thing that it produced was this acetyl-acetyl-CoA, which will go on to make ketone bodies. And it can go into the TCA cycle to produce 20 ATP if it was catabolized. Okay. There was this additional 2.5 that got made here in this step, which we accounted for. And I would tell you on a test that it would make 2-glutamate as well, so that you could take into account this this oxidative deamination. Okay. So let's just do the other one. Okay, where am I over here? Okay, let's do leucine and isoleucine. Okay, so leucine has 6 carbons and is catabolized in multiple steps. So we're going to see that there's 3 carbons that are generated by acetos to form acetyl-acetyl-CoA, which is going to be in the pink over here. And we're going to have 2 carbons that are going to produce acetyl-CoA. All right, so you can see then that leucine is going to be a loser in that it is only ketogenic. It only produces acetyl-acetyl-CoA and acetyl-CoA. All right, so let's write this down. How many nitrogens does it have? Whoops. It has 1, so you're going to go 1.5 in the urea cycle, and then 2.5 going through oxidative deamination. Okay, we know that through leucine, there's going to be an acetyl-CoA that gets produced and an acetyl-acetyl-CoA. And we call this ketogenic. Okay, so if it's acetyl-CoA, there we get 10 ATP being produced. And we also get 20 ATP from this acetyl-acetyl-CoA. So when we add this all up, we'll write this down in green. We're going to get plus 31 ATP equivalents. Okay, let's try lysine. Okay, so lysine has 6 carbons. 1, 2, 3, 4, 5, 6 has 2 nitrogens. So we have 2 nitrogens there. That's going to cost you -3. And again, we're going to get +5 here because we're going to get 2 glutamate produced. And I would show you that on a real exam too, okay? So we get lysine being used, and it's going to produce only acetyl-acetyl-CoA. So all of these carbons on here are just going to go on to make this acetyl-acetyl-CoA. That's the main thing I want you to get across there. So acetyl-acetyl-CoA. We call this strictly ketogenic because the only compound that it made was acetyl-acetyl-CoA, which has no potential to make glucose. So this is going to make 20 ATP. And then net-wise, we have to take into account that we have +2.5 again from this alpha keto adipate. Okay, so net-wise, this means we're going to have 24.5 ATP. All right, so there's all the ATP that we get out of there. And again, if I gave this to you as an exam question, it would show you all this, right? It's basically showing you the lysine. You just follow the arrows. It's taking it down to alpha ketoadipate, which will make an NADH here, to gluteryl CoA, which will go through 4 steps to make this. And then you'll know that that is 20 ATP worth there, okay? All right, the last one is isoleucine. So let's do that one. And isoleucine is down here. So we see from this that you get 3 carbons generated as propionyl CoA, and you get 2 carbons going into making acetyl CoA. So let's write down the in the table here. How many nitrogens does it have? It has 1. So you get minus 1.5. And you're going to get 2.5 produced through oxidative deamination. We produced an acetyl CoA. And we also produced a propionyl CoA. So we classify this as being both, because this compound over here is ketogenic only, and this compound down here is glucogenic, because if you remember from odd chain fatty acids, it gets converted back into succinyl CoA, which can go into the TCA cycle. So this produces 10 ATP, this produces 4 ATP, and then when we add that all up together, we're going to get a net of 15. All right, so that is it, okay? So let's answer question 8 down here. It says, where have we seen propionyl CoA before and how does it enter TCA cycle? Right, so propionyl CoA is from odd chain fatty acid oxidation. And remember that it enters the TCA cycle as succinyl CoA. Okay, and it makes it glucogenic because it enters in at the bottom of the TCA cycle, like right here, which has the potential to make oxaloacetate, which can go on to make glucose through gluconeogenesis. That concludes our discussion for set 8, amino acid catabolism, fate of carbon skeletons. Stay tuned for set 9 on amino acid synthesis.